Good afternoon. Today's lecture and our next lecture deal specifically with putting all of the pieces of the puzzle together that we have learned about from the beginning of the semester to this point. We have discussed extensively the innate immune response, the adaptive immune response, and lastly, we discussed the complement system. Now what we're going to do is look at individual categories of pathogens, how the host immune system responds to those pathogens, as well as discuss evasion mechanisms that pathogens have evolved in order to get around or away from our immune response. At the beginning of the semester, we discussed the fact that a pathogen is an organism that causes a disease. Pathogenesis is the means by which the disease-causing organisms attack the host. We discussed then that there were four broad categories of pathogens. There were viruses, bacteria, fungi, and parasites. We have spent most of the time this semester talking about bacterial infections and a little bit about viruses, but with today's lecture and the next lecture, we are going to look at all four categories and the immune response, specifics in the immune response that deal with those pathogens. Both the innate and the adaptive immune response, as well as the complement system, do in fact provide defense against pathogenic organisms. However, there are millions of deaths worldwide due to infectious disease. Infectious diseases are caused by pathogenic microorganisms, and the diseases can be spread directly or indirectly from person to person. In this particular figure on the left-hand side, what you are looking at is the number of deaths due to specific causes. And what you can see highlighted is the piece of the pie referring to infectious disease. One of the things I like to highlight with this particular figure is that the pie piece right next to that is labeled neoplastic diseases. These are cancers. And so oftentimes we think about cancer as being such a huge component causing death worldwide. And what you can see is that infectious diseases actually cause more deaths than cancer do worldwide, particularly in third world countries. In addition, you can see in the table on the right-hand side of this slide a little bit of a breakdown as far as what the infectious disease is and the number of deaths annually that are caused by those particular infectious diseases. Interestingly, and quite timely with what's going on today, respiratory infections are the number one cause of deaths annually as far as infectious diseases are concerned. So let's spend just a little time talking about pathogens in general and the location of where they can actually interact with the host system. The first thing we discussed back at the beginning of the semester was that in order for an infection to take place or to occur, there needs to be a breach in a physical barrier to allow for the potential pathogen to enter into the host system. What you can see on the left-hand side of this particular figure are entry points for pathogens to enter into a typical human host. On the right-hand side, you can get a little bit closer view at the tissue and cellular level as far as where pathogens can in fact occur. You can have pathogens that are extracellular, meaning that they occur in the tissues of the host and don't actually enter into cellular components. These would be pathogen categories such as viruses, bacteria, parasites, and fungi. So all four of those categories are represented and can be extracellular. In addition, you can have pathogens that actually enter into the cell. In this case, the types of pathogens that you can find intracellularly are viruses, bacteria, and parasites. Now, this slide is really just meant to be Okay, so now let's delve a little bit deeper and talk about specific pathogen categories. The first category we are going to discuss today are viral pathogens. As we have discussed in the brief sense throughout the entire first half of the semester, is that viruses need to infect a host cell. Once they get inside the host cell, they take over the replication, transcription, and translation machinery to make new viruses. Viruses cannot live on their own and rely on host cellular machinery to replicate and make new viruses. When summarizing viruses and viral structure, we can talk about the two main components found associated with all viruses. All viruses have some sort of nucleic acid as their genetic material, either DNA or RNA, as well as having some coat that is typically a protein coat or a lipoprotein coat that encapsulates all of the genetic material. Now, there are a variety of viruses that are much more complex than the simple description I've just given you, but for our purposes, we're going to look at very basic components of viruses. And what you see in this particular slide are just different examples of viruses that have been identified. There are, in fact, viruses identified to infect just about any cell type. The very first one is tobacco mosaic virus, which obviously infects plants. Adenovirus and influenza viruses are things that are very common in humans and other mammal systems. And then lastly, you have an example of a virus that infects bacteria. So again, just about every cell type has been identified to be infected by some type of virus or to have the potential to be infected by some type of virus. Now we can look at one specific type of virus, the influenza virus, and look at it in a little more detail. This particular virus is known to come in 3 different or basic types, type A, B, and C. All of the influenza viruses do in fact have 2 key viral components that are glycoproteins, which just means they're proteins that have a carbohydrate linkage associated with them. The 2 key viral glycoproteins associated with influenza is hemagglutinin, abbreviated HA, which is a component that allows for the attachment of the virus to the host cell, and neuraminidase, abbreviated NA, which helps new viruses escape from the host cells once new viral particles have in fact been synthesized. This particular virus is known to have RNA as its genetic material, and you can see in the figure that this particular virus is a little more complex than the simple example we talked about on the previous slide. Now, the reason I wanted to talk a little bit about influenza virus is to discuss in general how viruses do in fact enter into a host cell and how they take over the replication, transcription, and translation machinery to make new viruses. So again, this example is specifically for influenza A, but it does give you a general idea of the viral infection process. So in very general or simplistic terms, step 1 of the viral infection process is that the virus needs to enter the cell in some way. This typically occurs by interaction of cell surface receptors on the host cell that do in fact have an affinity for viral components. And it is this interaction between the viral component and the cell surface receptor of the host cell that typically allows for the entry of the virus into the cell. Step 2 would be referred to as unpackaging. The genetic material of the virus does in fact need to be unpackaged from the viral coat so that that genetic material can be accessed and utilized in the production of new viral particles. Typically, step 3 involves some component of viral genome replication. Now, this particular virus is an RNA-based virus, and so there are additional steps that are required for this replication process. But for now, we'll just simply state that the viral genome does in fact get replicated. The next step typically involves transcription of viral genes. Once those viral genes are transcribed, the host ribosomes are utilized in the translation process to synthesize viral proteins. Once those viral proteins have been synthesized, the next stage in this process is to produce new viral particles, basically package all of the genetic material in with the proteins that have been synthesized. And then lastly, there needs to be release of the virus. These new viral particles can go and infect new cells. Okay, so how is it that the host immune response can fight a battle or an infection against a typical virus? What we're going to talk about next is obviously not an all-inclusive list, but just a highlight of some of the components that we have discussed as far as immune responses to viruses. There is in fact an innate immune response. In fact, if you recall, we discussed the toll-like receptors back at the beginning of the semester, and the toll-like receptors were PRRs, pattern recognition receptors, and their job, one category of toll-like receptor, is to recognize viral components. When those viral components are recognized, they induce expression of specific types of cytokines, which include interferons. Interferons are one class of cytokines that definitely play a role in inhibiting viral replication, and we'll look at that in just a minute on the next slide. In addition, these cytokines that have been initiated due to the detection of a viral pathogen also initiate natural killer cells and their lytic activity, which means those natural killer cells, again, destroy those altered self cells that have been infected with the virus. There is, of course, also adaptive responses that occur in response to viral infections, both humoral and cell-mediated. With the humoral response, there are plenty of different antibody classes that do, in fact, play a role in fighting off viral pathogens. For example, IgA, which is the secretory class of antibodies, are released to then bind to viruses to prevent actual infection or entry of the virus in the host system. IgG and IgM play a large role in opsonization. Opsonization, again, tags a pathogen for destruction via phagocytosis. We know that IgM also plays a role in neutralization of viral particles. And so you can see that there are plenty of examples of humoral responses that are initiated in response to viral infection. There are also several examples of cell-mediated responses. The obvious, which we've spent some time talking about, are the cytotoxic T lymphocytes, whose job it is to destroy altered self cells that are infected with a virus. In addition, we also know that helper T cells play a role in allowing and initiating natural killer cells to do the same via ADCC. And lastly, not listed on this slide, we know that the complement system is also activated specifically due to interactions between IgG and IgM to activate the classical pathway. Okay, now let's look with a little more detail and specificity of what happens when a specific type of cytokine known as interferon alpha or interferon beta, in this slide is abbreviated IFN. So again, this is a specific type of cytokine that is initiated in response to viral infections. What you see in this figure is the cell membrane of a host cell that is infected with a virus. There is an INF alpha and beta receptor that is part of the cell membrane structure of the host cell. Once the interferon, the cytokine, has been produced, the cytokine is the ligand that binds to the receptor. This then signals the cell to act in a specific way, not unlike all of the receptor-ligand interactions we have discussed all semester long. Specifically, what happens in this interaction is two alternative pathways. The pathway on the left essentially activates an enzyme known as RNase L. RNase L is a ribonuclease, and ribonucleases are general categories of enzymes that specifically degrade RNA. So in this pathway, this enzyme is activated and it triggers the degradation of messenger RNA in the cell. On the right-hand side, you also see a signal transduction cascade. But what I want you to take away from the pathway on the right-hand side is that a specific protein, EIF2-GDP, is made non-functional. This is a protein that is required for protein synthesis to occur. With both of these mechanisms, the end result is the inhibition of protein synthesis in the host cell, either by destroying the mRNA, which acts as the template for protein synthesis, or by making a component of the protein synthesis machinery non-functional. Either way, protein synthesis is inhibited. By inhibiting protein synthesis, all activity is stopped in the host cell in hopes of not allowing for viruses to be replicated. Okay, so now at this point, what we've done is reviewed just a bit what the host immune response is in response to general viral pathogens. What I'd like to do is turn the tables just a little bit and talk about how viruses have evolved to evade the host immune response. I'm going to give you several examples here just to give you an idea how this evasion mechanisms do in fact take place. One example is hepatitis C. Hepatitis C is known to produce a protein that inhibits one of the steps on the last slide that we just discussed. So by stopping that signal transduction cascade, protein synthesis does not get inhibited, which allows for the continual production of viral components. Another example of viral evasion is from the herpes simplex virus. The herpes simplex virus produces a protein that inhibits TAP. Now, TAP, if you go back and recall, is essential for allowing for our viral antigens to be displayed to a cytotoxic T cell. So if that step is inhibited, the antigen doesn't get displayed, and hence that mechanism of recognition and interaction by a cytotoxic T cell does in fact not occur. Herpes simplex virus also has the ability to inhibit C3b of the complement system. One last example of viral evasion comes from the adenovirus. Adenovirus has a protein that is able to reduce the expression of class 1 MHC in the host. By doing that, that results in little to no antigen presentation, hence no destruction by cytotoxic T cells of that target cell. Okay, the next category of pathogen that I want to discuss are bacterial pathogens. Now, we have spent quite a bit of time using bacterial pathogens as examples with all of the mechanisms that we have learned, but now we're going to, again, put all of those puzzle pieces together. So just to recap a little bit, bacterial pathogens are in fact prokaryotic cells. They are typically smaller than eukaryotic cells. They lack internal organelle structures that are surrounded by membrane. They also lack a membrane-bound nucleus. And bacteria also have rigid cell wall structures. There are lots of different types of bacterial pathogens, and obviously the list would vary depending upon the specific type of pathogen we would be discussing. With bacterial infections, there are 4 primary steps involved. These include the attachment of the pathogen to host cells, proliferation of that pathogen. Once proliferation has occurred, invasion of the host tissue by the bacterial pathogen, and lastly, toxin-induced damage to host cells. This again allows for the infection to take place. So for step 1 in bacterial infections, the attachment of the cells to the host cells, this is blocked by the production of host IgA. The IgA is the secretory antibody, and the secretory antibody being produced should bind to the bacterium and neutralize the bacterial pathogen and not allow the bacterial pathogen to enter. There are some bacterial pathogens that produce enzymes known as proteases, and proteases are enzymes that degrade proteins. Specifically, some bacteria produce proteases that cleave the IgA dimers. By cleaving the IgA dimers, the dimers are made ineffective, and hence the bacteria has now the ability to attach to host cells. Another host immune response against bacterial pathogens is phagocytosis. In particular, opsonization by antibodies as well as C3b play a really big role in targeting bacterial pathogens for phagocytosis. There are bacterial pathogens that have surface antigens that are able to inhibit phagocytic cells, hence stopping phagocytic activity and not destroying that particular pathogen. And lastly, this slide puts those puzzle pieces together again in just a little different way. So we know that there is a breach of the physical barrier that needs to occur that will allow a bacterial pathogen to enter into the host tissue. Many bacterial pathogens do in fact produce toxins that promote infection by damaging host cells. Some of these toxins are able to actually disable immune cells from functioning properly. What we know does occur in response to an infection is that there is toxin neutralization that can in fact occur. There is also complement-mediated cell lysis that takes place, again specifically targeting cells for phagocytosis. Opsonization and phagocytosis in response to antibodies that can specifically bind to the pathogen. We also know that components of the complement system initiate production of inflammatory response components. And we also know that there is extensive amounts of cell migration from the host bloodstream to the target location due to cytokine production. I think we've spent the most time this semester talking about bacterial infections and the response of the immune system to those bacterial infections. So this is really just an overview of what we discussed all semester long. In our next lecture, we'll look at the final two pathogen categories, parasites as well as fungal pathogens.